Okay, so welcome. I'm really glad to have all of you here with us today. My name is Elizabeth McMentatenko, and I'm a librarian here at UC Merced. And I'm really pleased to be able to introduce Dr. Sora Kim, who's going to be giving our Library Faculty Author Series talk today. The Faculty Author Series showcases the latest research and scholarship by UC Merced faculty members. So if you or someone you know would like to be featured, please let me know. For today, we're thrilled to have Dr. Kim here with us. Dr. Kim holds a PhD in Earth Science from UC Santa Cruz, in addition to a BA in Earth Sciences and Environmental Studies from Dartmouth. She was a postdoctoral research associate at the University of Wyoming and has previously worked as the T.C. Chamberlain Fellow at the University of Chicago and also at the University of Kentucky. Much of her recent work has focused on sharks, but she also has ongoing projects about mammals, nutrient cycling, and lake sediments. And she recently received a career award from the National Science Foundation and has been featured on Shark Week. So we're really happy to have her here with us today. I'll turn this over to you.
Thank you. Hi, everyone. Thanks so much for coming. I forget what I said that the title of my talk would be, but then I was on the airplane on Monday and I was like, oh yeah, I can do the Secret Lives of Megalodon, recordings in tooth chemistry. And I was like, maybe some people will get it with the Secret Life of Bees, but maybe it's too far. Here's just some images of recent work with Megalodon, and then a shot from when I was featured on Shark Week. We were recording, I think that was in February, we did the recording for that, and that has gotten a lot of hype here at the university. So I wanted to start off in sort of thinking about, why do we care about sharks? I mean, sure, they're featured on Shark Week and there's Shark Fest. It's the only organism to have, 2 weeks of popular programming dedicated to it that the public tunes in for. But there are some reasons beyond just the entertainment value of sharks. And there has been some severe declines in shark populations due to human impacts. Part of this is fishing pressure. There are 4 featured sharks here: the hammerhead sharks, whaler sharks, tiger sharks, and white sharks off the coast of Australia. And here is the timeline from 1960 to 2020 and the catch per unit effort. So sort of the abundance of sharks that are caught, given how much work that they're putting into it. And there's a lot of noise in the data, but to take away from it is that all of these lines are decreasing. There are declines of up to 92% in the population numbers of these various sharks. And so these sharks are, these larger charismatic sharks that sort of people seek out and like to see in the waters, but overall they're all declining, which is problematic. And there's actually 500 species of sharks. And so there are a lot of other sharks that we don't keep track of, that we don't know as much about, that are also likely facing these types of declines. And one thing that maybe is a lot harder to capture is actually how modern climate change is affecting marine ecology. So here I have an image by Ray Troll. He's an artist and paleontologist, but really obsessed with fish and sharks. He lives in Alaska. So I'm featuring a lot of his artwork with permission here. And the reason why I think that his work is really interesting and fascinating is because he actually does the same thing that I do scientifically in his art, which is going sort of back and forth between the modern and the paleo. And we really need to look at the paleo in ancient times, because there's a lot that we don't know about the modern ecosystem still. We can do things like from satellite images, look at sea surface temperatures and get at how there are deviations from normal based on these satellite images. We can also record things like net primary production change. And so that's looking at sort of chlorophyll concentrations from satellite images. So we can look at all of these things, but in reality, oops, I pushed the wrong button. But in reality, there's a huge part of the ocean that we don't know very much about. So if you this is just a table of how sea surface temperature and net primary productivity interact with one another. And so we have these interactive effects for most of the ocean, but here at high latitudes in the Arctic and the Antarctic, it's a blank slate. We don't know very much. And those are areas, especially that sharks today, not very many of them live in, but also climate change is impacting those areas at a much higher rate in terms of time and number of species. So there's an importance of looking at the fossil record that may have some clues. And sharks represent a very successful evolutionary strategy within the fossil record. They have a 400 million year fossil record, and they've survived through 4 mass extinction events, which are shown here in the stars. So here's a depiction of the geologic time scale, also by Ray Troll. And sharks sort of came to be during the Silurian. That's where we have the oldest fossils from them, about 440 million years ago. Dinosaurs are in this green area. And so sharks came around like, 200 million years before dinosaurs. And even though the dinosaurs were taken out by the meteor at the KT extinction, sharks continue to today. So they're incredibly resilient. In fact, you can see this is sort of, these are called spindle diagrams showing like the abundance and the diversity of different shark taxa. And sharks have this one bottleneck here at the Permian-Triassic boundary. That took out like something like 75% of all life. It was a big one. But otherwise, sharks have been fairly unaffected by all of these mass extinction events. They have a success record in the fossil record. And it's kind of amazing that they have found a way to make a living. And probably the most famous ancient shark of them all is megalodon. I have some depictions here. They just recently released Meg 2, and so there are a variety of shark movies, but I feel like the Meg franchise is quite big. The average megalodon adult that we think, based on sort of estimates on how big the vertebrae are in the teeth, is about 50 feet. So that's a little bit shorter than 2 school buses backed up end to end, so fairly big. The only shark that we have that's even on the same order of magnitude today would be a whale shark, but whale sharks are usually, they top out at like 30, 35 feet. So much bigger. A great white shark is depicted here as 15 feet. So it makes the great white shark look itty bitty. And then this is sort of an image that's classic of showing the size of a white shark jaw compared to the jaw of a megalodon. And if you see like old timey photos and stuff too, you'll see people like sitting inside or standing inside of a megalodon jaw replica. And then this was an artistic rendition that was done for a recent paper of mine. And so we do think that given megalodon's size, that they probably ate marine mammals, or that they were apex predators, but that is a big question, as well as like, why did they go extinct? So I was fortunate to get a National Science Foundation grant to be able to sort of look into and delve further into megalodon ecology. So today, just to give you a roadmap of what we'll be talking about, we'll be looking at what I call biological glimpses through chemistry. I think that oftentimes people think of me as being like a paleontologist or marine biologist, that I'm like Indiana Jones style, like out doing work, like excavating fossils in the desert or in a shark cage, but that's not it at all. I do a lot of chemistry essentially. So I'll give you a short review of isotopes, the main methodology that I use. And then a few questions that we'll explore is, how do megalodon compare to other sharks on the food chain? Are megalodon warm-blooded, and why does that matter? And how do these new insights change our ideas of megalodon extinction? And some images of megalodon shark teeth compared to the size of various hands. There's also an exhibit that features megalodon, a life-size sculpture of megalodon in the Smithsonian. I think there's one at Cal Academy right now too, that I need to go check out. And then another movie poster for the back. So Earth's climate has changed over this last 65 million years, and this is all recorded in stable isotopes. So anytime that you sort of hear on the news or hear references to climate change over deep time, this is all based on isotopes. So isotopes can tell us about climate from about 60 million years ago in sediment cores taken from the deep ocean. And then in thousands of years ago, these isotopes are captured in ice cores. And then in historical time, I mean, we just have instruments that measure it. And then these are sort of climate projections of where we could go. But what I would like you to note is here, 0 is pinned at the average temperature between 1950 to 1990. And you can see these wild fluctuations that have actually happened in the last 65 million years. I mean, during the Eocene, which is some of the warmest time recently, we had temperatures between 10 to 15 degrees elevated of what it is, you know, from these historical times. And then even during the slow decline, I mean, this decline is happening over almost 60 million years. And then the climate change that we're seeing today, you know, is here. And this is just in years, it's not even millions of years. And you can sort of see the trajectory for that climate change that's happening. My computer is doing an update. So this is the time that megalodon existed during the Miocene to Pliocene. They evolved about 22, 23 million years ago based on their fossil record, and they went extinct about 3 million years ago. So their early part in the Miocene was a warmer time period, and then there was this transition to cooler temperatures. So one of the hypotheses about why megalodon went extinct was that there was oceanographic changes that actually happened during this time that caused restructuring of maybe food webs, of ocean currents, and that megalodon was not able to respond to those changes. So I'm going to take you back to like high school or gen ed chemistry and talk about what is an isotope so that you have some sense of going forward and looking at some of the data. So an isotope is an atom with the same number of protons because that's what determines its element, but there's different number of neutrons. And so it doesn't actually have a charge to the particle, but it creates extra sort of mass within the atom. So for hydrogen, we have hydrogen 1, hydrogen 2, and hydrogen 3. And this changes where you have one proton, but there's different numbers of neutrons. And then in carbon, we have carbon 12, which is the stable and more abundant form. Carbon 13, which also has 6 protons, but 7 neutrons and is also stable. And then carbon 14, you might be familiar with, because it is actually a radioactive decay, and that's how we do a lot of dating for things that are, I think it's usually about 25,000 years old or younger. You can do C14 dating. And so these fossils are millions of years old, so we don't necessarily use C14 dating, but that's a term that a lot of people are familiar with in terms of isotopes, but we don't look at these radioactive isotopes, we look at the stable isotopes. So why does it actually matter in terms of this extra neutron? The small difference in weight can cause differences in sorting between the light and heavy isotopes of various reactions. So these can be biological reactions. In this example, it's carbon, that you have some substrate. You can think of it as food that you're eating. And then there's the metabolic processes that are happening in your body to convert that to energy. And the C13, because it's a little bit heavier, it forms stronger bonds, or it can just take more activation energy to get it over this energy hill that's needed to create a product. The product being maybe like your muscles or your bones and things like that. So that's sort of what's happening in the reaction process. Another example that might be a little bit more relatable is the evaporation of water. So evaporation actually selects for the lighter isotope. You have a water body, like the oceans, for example, and when you're evaporating the water molecules, the water that contains the light isotope, so oxygen 16, it's gonna actually evaporate faster than the oxygen 18. So then the water vapor, like the clouds that are over the oceans, they actually have more oxygen 16 in them than the water that they were sourced from. And then in the corollary that you have these clouds then with water molecules, but these heavier isotopes, they actually condense faster and then they precipitate out faster because it's a little bit heavier. And so in terms of bond strength and stuff. So rain is actually isotopically heavier than the vapor that it comes from. So these are just examples. I explore some of this with my research, but we're not going to talk as much about it. But I am going to be showing you a lot of isotope data because that's how the main form of my scientific research. So I just thought I'd give you a little primer on it so that you weren't like wondering the whole time, like, oh yeah, what was an isotope? So that's enough of that. In terms of my applications, one isotope that I look at is carbon isotopes. It varies with ocean productivity, and this is related to carbon dioxide uptake by primary producers. So these can be foraminifera, diatoms, and algae in oceans. This is a chlorophyll concentration satellite map where red has the highest amounts of chlorophyll. So here you can see that there's a lot of chlorophyll around Alaska. So this would be a satellite image of the summer, of the northern hemisphere summer. And then magenta is where chlorophyll is the lowest. So this chlorophyll gradient actually causes this gradient in carbon isotopes with latitude and also nitrogen isotopes. But then we also have offshore, nearshore gradients where you can sort of see, especially with the upwelling that's happening here along the West Coast. So this is probably happening differently in past oceans, but isotopes can really give us a way to explore these kinds of dynamics and have these ideas of what is happening in terms of ocean productivity of the past. Oftentimes, aspects of diet are of interest as well and recorded in isotopes. So this is a complex marine food web, and you can sort of see how everything is linked to one another. And with isotopes, we usually collapse this down to sort of a food chain and think about trophic levels. So in terms of trophic levels, we have the producers, the primary consumer, secondary consumer, tertiary consumer, and then the apex predator. And nitrogen isotopes, it actually increases as you go up this food chain or up the trophic level. And this is because the waste products have more of the heavy nitrogen. So this is an image of a horse, but you can also think of our own bodies. The way that you take in most of your protein, your nitrogen, protein is what contains the nitrogen, is through the meat that you eat. So there's nitrogen in that protein. Your body does stuff with it, creates more muscle mass, creates bones, but you are losing some of that nitrogen either as feces, poo, or urine and pee. So what's leaving your body is actually oftentimes the lighter nitrogen because it can get through that activation energy much quicker, and then your body stores the heavier nitrogen. So as you go up in trophic level or up the food chain, your nitrogen isotope levels are increasing. And so even for us in this room, if we were to take our fingernails or our hair and analyze the isotopes in them, you would be able to see sort of like who has more, who is higher on the food chain than other people, which is an exercise I do in my stable isotope class with students. That's fun. So that was the review of isotopes. Now we're going to look at how do megalodon compare to other sharks on the food chain. So a small plot twist in being able to apply this whole isotope method is that nitrogen is in protein. Protein's an organic material, and it doesn't actually preserve very well. It's a soft tissue, actually. So how are we actually going to learn about diet? And so here I've highlighted some protein structures or some different materials that contain protein. And the longest that they preserve, is less than 1 million years. So when I was a graduate student, this is when we actually had landline phones. So we had one in the lab, and a reporter called the lab and asked to speak to me and was like, okay, like you do this isotope thing with sharks. Like if I send you a megalodon tooth, can you tell me what it ate? And at the time, we didn't have these new methods that we've developed recently. And I was like, no. He's like, but you could be on the New York Times if you could tell me what a megalodon ate with isotopes. I was like, I can't do that. Like that would be lying, it would be cheating. So, but we've made some progress on that. So this is really work that was spearheaded by Dr. Emma Cass. She is now a postdoc at the University of Cambridge, but it was part of her PhD work. And it turns out that there are trace amounts of nitrogen bound to mineral. So on your teeth, we have enamel, and that enamel is actually a mineral matrix and very resistant to decay, and it would fossilize very well and all of those things. But there are actually trace tiny amounts of nitrogen in there because of how enamel forms in terms of there's a protein matrix goes down and then is reabsorbed, but not all of it's absorbed. So we have this enameloid bound nitrogen isotopes. They've also done it on cows and also foraminifera, which are like inorganic, like tiny little microorganisms in the oceans. So the first part that we did was like, okay, like, great, we can measure this enameloid bound nitrogen, but how does it relate to the nitrogen that we would measure in modern shark teeth? So here on the x-axis, we have the nitrogen isotope value of the dental collagen. So that's the inside part of your tooth versus the enameloid bound nitrogen. And then this is the 1-to-1 line, that's the dashed line. And we found that it was actually elevated. So it's like, oh, okay, these aren't like related directly. We have to account for this offset in this enameloid bound nitrogen. And so this is the offset. And then we were interested in looking at these megatooth sharks. So within the line of megalodon, there are other megatooth sharks where they have this really broad shape to their teeth. And the earliest ones sort of date back to, this is actually just showing piscivorous sharks. Sorry, I got ahead of myself. I got too excited. So in the gray dots here, these are piscivorous sharks, so sharks that are eating fish. Run-of-the-mill sharks, you can see that they have, a very constant nitrogen value through time from 65 million years ago to modern. This is showing megalodon in the orange, and then white sharks and the proto-white sharks in like the light blue and yellow in the Pliocene, so about up to 5.3 million years ago. And then here are the lineage of Ototus. So these are the megatooth sharks that have like the really broad teeth that are very flat. And so they originated here, in the Paleocene about 56 million years ago. And you can see that they have this elevated nitrogen value. And this is even before we have like actual megalodon teeth from the Miocene. So it looks like that they developed this ability to eat at really high trophic levels, even before their size got big. And then this is the ancient sort of the basal taxa for megalodon. It looks like it ate fish. And then I have realized that it's really hard for people to digest scientific figures oftentimes. So I actually work with a scientific illustrator who worked with me as an undergrad. She helped me keep sharks in captivity. And it's great because she's like actually observed sharks and stuff. And she made this really nice diagram. And so this here is actually showing the offset in nitrogen isotopes from the fish-eating sharks. So if we have fish-eating sharks for that same time and place, and then we compare like what this the megatooth shark would look like. So here is Ototus megalodon, our megalodon shark. It's actually not at the highest trophic level. This entire sort of lineage of Ototus, some of them are actually at a higher trophic level than even megalodon. So we were kind of like, oh, well, that's interesting that, that the size followed the diet. And I guess that makes sense that you have to be able to like, get those resources and have the diet in place before your body shape can actually change, or else you can't get the fuel to keep the engine going. Another potential trophic level indicator that we found is zinc isotopes. And this was a paper that was published in 2020 with mammals. And they found that as you go up in trophic level for mammals, this is like a cow, a warthog, and a puma type of animal, that we have this decline in zinc isotopes. And zinc is really well preserved in the fossil record. It's preserved as it's necessary for biomineralization. And we compared, modern tiger sharks to early Miocene tiger sharks. And the most important thing to take away is that these images look very similar. So there's not a lot of alteration that's happening. And then when we looked at, different parts of the tooth and how much zinc existed and what that zinc isotope value was, we found that here at the tip, the enameloid is actually the thickest at this point, and it looks like we have the most amount of zinc. And so we're saying that that zinc isotope value is probably best preserved. And this is work done by Dr. McCormick, who's at Frankfurt University. And so we took sort of a similar approach to the enameloid-bound nitrogen, but we actually did a lot more samples of modern sharks to compare with the zinc. So zinc isotope values in modern shark teeth, they reflect trophic level and are similar between localities. So these are all the different modern sharks that we looked at in that nitrogen study. We actually only looked at one species. And then we sort of organized them based on like what trophic level that sort of shark ecologists think of. And we're like, oh yeah, this is pretty constant. Like these orange ones belong, oh, I can't even read it, but the colors are supposed to be similar families and stuff. So these orange ones are sort of similar to these orange ones. These red ones are similar to these red ones. So it's like even between the different places that they look like that they have more consistent values. And so one of the problems that we have with using carbon and nitrogen isotopes is that there's a lot of spatial variability. I showed you that that chlorophyll concentration map of the Pacific Ocean, and there's all these baseline differences. So it would be cool if we had a way to look at trophic level that we didn't have to count for all these baseline differences. So that's our hope with zinc isotopes. We've also analyzed ancient shark teeth, and we found that modern and ancient shark taxa have actually very similar zinc values, which suggests that these ecological roles are conserved through time. So, for example, we have, this is a tiger shark. So this is a modern tiger shark, and then this is an ancient tiger shark. And you can see that their values kind of overlap with one another. So it means like a tiger shark of the past is doing a similar thing to tiger sharks today. Here are sand tiger sharks. And again, we have two modern sand tiger sharks from different localities. And then these are ancient sand tigers. So maybe more variability in the ancient, but at the same time, it looks like there's a lot of things that, that they're probably eating similar things, have a similar type of lifestyle. And then when we get to white sharks actually have a high trophic level today. We know that in terms of what we observe them eating, but we also see that for the Miocene and the Pliocene. So this is today's white sharks, and then this is the prototype of the white shark and ancient white sharks. And it actually looks like white sharks today might have a higher trophic level than the white sharks of the past. There's some individuals that are high. We oftentimes, I think of animals as being very monolithic, that like all the animals, like white sharks do this thing of eating seals, that's what they do. But in reality, like sharks have dietary preferences, different animals have dietary preferences as well. So you can imagine that there are like some that are like picky eaters and others that are less picky, and they might be doing different things. And then finally, to highlight the megatooth sharks, they fed at a high trophic level. So here's megalodon right here. But you can see that there's a lot of variation. We actually measured a lot more megalodon teeth than other shark teeth because they were the feature of our study. And we were really surprised by the high amount of variation in megalodon diet. but that's not like what gets picked up by news agencies and stuff. They really like to harp on just like how high up on the food chain and that they're apex predators. And then we actually see overlap between megalodon and white sharks in the early Pliocene. So this is kind of the time that that megalodon is maybe potentially getting close to going extinct in the Pliocene. And we see this overlap between megalodon is in blue, and then white sharks are in the purplish color. So this really sets up this possibility that they are fighting for the same resources. Maybe they're not like coming face to face in a fight, but their food source is still very similar, and they might be occupying like similar parts of the ecosystem. And then these are other white shark and megalodon parents. So then, next part is, are megalodon warm-blooded and why does it matter? So shark teeth also, they record the water temperature within their mineral structure. This is sort of the chemical structure of enameloid or enamel. And I just highlighted the parts where there's oxygen within the structure of the system. I usually measure this oxygen within the phosphate. This is kind of like more traditional ways of going about analyzing isotopes in fossils, and what I was more trained to do rather than the newer fangled fancier enameloid bound nitrogen and zinc. And we do these measurements here at UC Merced. But what you can see here on the x-axis is the offset in oxygen isotope values from this phosphate versus the water that they're living in, versus temperature. And these are some modern sharks, and you see that it plots very nicely. This is really related to like the source water of forming the enameloid is body water, and for sharks, that's the water, the ocean that they live in. And then the amount of isotopes that are incorporated into the mineral structure is temperature dependent. So that's again that energy hill that I showed you with the carbon isotopes. The problem with this, though, is that the shark teeth, they record the temperature, the mineral structure, but it's really dependent on the water. And so you might imagine for water that's dating back thousands of years ago or millions of years ago, tens of million years ago, we don't have great idea of what the oxygen isotope value is of the water. So that's been like a major obstacle for being able to do this type of work in the past. And so if we do compare this oxygen isotope value from megalodon versus other sharks from the same time and place, this is what the graph looks like. And you can see general patterns that these ectotherms, which are most sharks are the same temperature of the water that they live in. They're in blue. You can see some variation depending on where they're living in the water. White sharks are shown in the orangish color. So white sharks, they can actually elevate their inner body temperature by 4 degrees Celsius from the surrounding water. And then megalodon, we're just like, yeah, sure, we'll try doing this analysis. And they are actually quite warm. We're like, oh, okay, that's different. And then here in the burgundy is actually marine mammals. So we know that, whales, dolphins, mammals have elevated body temperatures. And so the megalodon's not quite as warm as a mammal, but it's still pretty warm. So this was like first surprise. And then we tried this other method called clumped isotopes. And so clumped isotopes are temperature dependent, but it doesn't matter the water composition of the substrate that's forming from. And the reason, well, the clumped refers to that CO2 has carbon, 2 oxygens, and it's clumping of the 2 heavy isotopes, so carbon-13 and oxygen-18. And it turns out that the amount of clumping that happens of these heavy isotopes being combined together increases, decreases, and then as temperature is low, you have lots of clumps. As temperature is high, you get fewer of these clumps happening. And so they looked at this for various natural systems and laboratory synthesized things, ranging in temperatures from 0 degrees Celsius all the way to 1,000 degrees Celsius. And this trend seems to hold. So we decided to do it with modern sharks first, and this is what our graph looks like. So most sharks are ectothermic and are the temperature of the water and the surrounding environment, and that's recorded in these clumped isotopes. So we analyzed a bunch of sharks. We actually do have one whale here, and found like that the line is very similar to what they had found in other biological organisms, but also just minerals as well, carbonate. And this was in collaboration with Dr. Mick Griffiths at the University of William Patterson. And these are some of the students. This is William Patterson University is a small, like, local commuter school in New Jersey. And they don't necessarily have a lot of lab and research facilities because it's like a teaching institution. So I hosted 3 of the students here in the summer of, I think it was 2019, to do some research. And it was cool that they were like, oh, these are like the nicest labs that we've ever been in. All of them have applied for graduate school. 2 of them are in grad school at this point, and it's something that like they were never really exposed to because they were all first-gen students. And then we were curious, do these clumped isotopes confirm this idea of the elevated body temperature of megalodon? So here's the temperature, and again, the megalodon is in red, and then mammals are in the burgundy color, and you can see that megalodon does have this elevated temperature, but not as high as the marine mammals. So what we came out of this with was that megalodon temperatures are elevated above modern sharks, but not as high as whales. So this is another artistic rendering where we have megalodon on top with the red, and then most like regular, or no, this is Carcharodon carcharias. So white sharks here, so you can see this offset, and then these are the marine mammals. So you can see how they compare in temperature to one another. So then finally, like, how do these insights actually change our ideas of megalodon? Like, who cares? Why is this important? I don't know that our knowledge of megalodon really makes a huge difference in our day-to-day lives, except that people are very curious about megalodon. But in the past, people, you know, their teeth are so big, they want to know what they were eating. And there's a lot of evidence of megalodon feeding on marine mammals with bite marks on bones. And a lot of this work is done in Peru, in this area, shown here in the map. And this is a paper where they did, I thought, a really nice illustration showing different parts of the bones and that where they have actually megalodon bite marks on them. So it's like, okay, like there's actually scientific evidence that megalodon actually ate marine mammals, not just like us wishing and thinking that this was the case. And megalodon was thought to feed on marine mammals, but in thinking about why megalodon went extinct, we thought that like, well, megalodon are big, they would need to eat large marine mammals, but it turns out the largest marine mammals are baleen whales, and they're at the bottom of the food chain really, because they're filter feeders. They're very low on the food chain. So this is a paper by someone named Nick Pineson, and this is actually looking at skull length and width to see how big the various marine mammals are. And so here, if you look, the biggest sharks, you know, where we have at the top of these graphs, they really come about more recently than back in time. And then these, at least marine mammals from panels A, B, and D, those are all very low trophic level. They're eating either plants or the sea cows up here at the top, and then baleen whales, which are filter feeders. So, you know, with our various isotopes where megalodon is at the top of the food chain, it doesn't really line up with the idea that they would be at the top of the food chain, but eating these large marine mammals. The other thing that was going on that I talked about before was that there was this shift in climate from the Miocene to the Pliocene. And so this could have really shifted like where their prey availability was. And when you're so large, you really require a lot to eat and that you're endothermic. And so we do see this change in marine mammal diversity at this time. So from the Miocene to the Pliocene, you can see for all of these different marine mammal diversity decreases during that time. So it does seem like that whether it was due to climate or some other factors that marine mammals became less abundant. And then our zinc and nitrogen isotopes are showing that they fed at high trophic levels. And then there's this idea of overlap between white sharks and megalodon. So if their prey, both white sharks and megalodon, are feeding at high trophic levels and they're looking for marine mammals, but the marine mammals are declining in abundance, there's going to be setting up some impacts of competition and resource use dynamics. So there's also, like I had mentioned, that when megalodon is so big and it has this elevated body temperature, it really comes at a high cost, needs to eat a lot more. There are advantages to being warm-blooded, that you can like dive deeper, you can swim faster, your muscles are capable of doing a lot more, but you have to eat a lot to compensate. So the show that I was on was called Jaws versus the Meg, and they made the case that they're both apex predators and they had this elevated body temperature, and they both had to deal with these changes in climate and prey availability and diversity. That's like what the science shows. In the actual Shark Week show, they actually had white sharks, or no, they had megalodon win because megalodon was bigger and all these, like they had the silly table. But I would actually say that white sharks won because they're the ones that are still around today and ultimately were evolutionarily successful. So I hope that you've learned a little bit about megalodon and maybe white sharks today. The last thing I would like to say that shark teeth are really abundant in the fossil record. They span, a lot of time, 400 million years, but also localities. So these are all the different fossil sites for sharks that are recorded in this database called the Paleobiology Database. And when you go into a museum, you can pull out drawers and they just have a box that has sometimes hundreds or thousands of teeth like this. And not very many people will actually utilize the teeth to study very much. And so this is kind of where my research takes advantage of this. And although we like to think about shark teeth as just being like these triangular pointy things, it turns out that there's a lot of diversity in the shark teeth. And I would argue that there's a lot more that we can learn about ancient sharks through the chemistry in their teeth. And these are just various shark teeth that are found in Florida. So it's not even a comprehensive diagram. So with that, I would like to thank various funding agencies, museums, and people that loaned us specimens, and thank all of you for coming to hopefully what you think is the cool shark show. Thanks.
So I think we have time for some questions. So I don't know, does anybody want to start with a question?
It doesn't have to be about the presentation.
I was actually going to ask about the specimens and the loans, or I guess in your scientific community, are loans very common, or is it quite a process to get them? And
they are common, but there is a process. There's usually quite a bit of paperwork that fills out, and you have to fill out, you have to give like your justification and the project idea. Especially with the stable isotope analysis, it's destructive. And so it really makes people kind of antsy. We started this project with megalodon, one, because people are really excited and hyped up about megalodon, but two, they have really big teeth. And so there's a lot of material for us to analyze with developing these new methods and stuff. But a lot of the shark teeth are very small. And so when we destructively sample them, that might be the entire specimen. So some of one of my ongoing projects is in Antarctica, and we have teeth that are 50 million years old from Antarctica, and we're destroying teeth, but before we destroy them, we like make casts and molds of them. We do really high quality 3D imaging of them. We thought about doing CAT scans of them, but then we were like, that's too much, like, that's a lot of time and money. We have to spend a lot of time in Berkeley to do that, but we really try to leave some sort of record for potential future projects. And with stable isotopes, I think that I've heard from museum curators that people will oftentimes destructively sample and generate the data, but then they'll never publish a paper. And so that's kind of a shame too, that you've destroyed the specimen, but then there's also like there's no data or results or anything to like look back on. And so a lot of museum curators are being much more careful and discerning about who they approve or don't approve. But there are like, I mean, there's hundreds of thousands, if not millions of teeth in museums. And I have young kids, and so the idea of me going on like a fossil expedition sounds really cool and interesting and fun and like field work. But in reality, with 2 young kids, it's like my husband would go crazy, like having to deal with my children for like 6 weeks while I'm on a fossil expedition or in Antarctica. So I oftentimes go to museums and utilize their specimens. And so I think that that's like also like a public resource that's available, and like people have already spent a lot of time and money collecting those samples and archiving them in a museum. So I think that they're a great resource.
And that's a really interesting connection between the museums and just like the scientific research, because I think even libraries, we think of museums more, not exclusively, but not sort of the hard sciences as much at times.
Yeah, absolutely.
I have a question about your slide where you had the map of where the teeth had been found. Do you know, or do you have an idea of, is there a reason why more teeth have been found in certain areas? Is it just people looking for them, or were there more sharks in that area?
A little bit of both. So in terms of the teeth are found in like sediments that used to be in the oceans, and so those where these are concentrated are like where those types of sedimentary formations are. So that's part of it, in North America for sure. But I would say that the lack of specimens here in the Southern Hemisphere is not necessarily completely driven by that. It is driven by one, a lack of fossilization, that there can be like really hot climates that would destroy fossils, or really humid climates, which would also destroy fossils. And then also the type of terrain, so that people aren't actually going out and looking for fossils in really difficult or in areas that are in conflict and things like that. And so the Southern Hemisphere is sort of notorious for being poor in fossil specimens, but I think that there are both sort of physical things that are going on related to that, but more importantly, there's a human aspect too. You had a question?
Yeah, I was gonna ask, what sort of dating methods do you use when something's too old for carbon 14?
Yeah, for a lot of these specimens, they might just be like looking at like this fossil community or microfossils, and if you find some certain combination of microfossils, especially for the oceans, there's an idea of like approximate what time period it's at. If there's volcanic ash, there can also be like uranium, thorium dating, or strontium dating that is there as well. There are also changes in sort of the strontium isotope values of the oceans based on like the age of rocks and things like that. And so we have actually this strontium curve through time, the oceanic strontium curve. And so you can also get the strontium value and try to like wiggle match it into there as well. So there's a variety of methods. None of them are super precise, like C14 dating, but there are some ways that we try to do it. But my project in Antarctica, it's like a huge problem. It's like, well, is this 30 million years old or is it 46 million years old? It's like 10 million years could mean a big difference.
I have another question.
Mhm.
So I know in one of your early slides, you had kind of a bottleneck where it looks like a lot of sharks had disappeared. Do you know what happened then or what? And you
Yeah.
said a lot of animals had disappeared at that
Mhm.
point.
Yeah, that's the Permian Triassic mass extinction event. And I think that there's still a lot of questions. I mean, it was a really long time ago. So there's still a lot of questions as to what happened, but it really seems like that mass extinction event really affected marine organisms really heavily. And so whatever it was, it took out, I think it's like 75% of marine biodiversity. Yeah, it was bad. Hopefully we will not repeat that. Yes.
I missed part of the intro, so this may have come up, but can you say a little bit just about like how you got into this field, or if you were, like you mentioned, the chemistry element, but like looking at shark teeth,
Yeah,
like, was that where you were headed, or did that
no,
sort of, like,
no.
come in at some point?
No, I came to college and I like wanted to do like, I wanted to work in environmental issues. Like my internships in college were mostly like, with environmental lawyers and environmental policy and things like that. So I guess I enjoyed things related to the earth and the environment, but then I became, an earth sciences major and I got introduced to this method of stable isotope analysis. And I just thought it was so cool that you could apply it to so many different types of systems. Like in my lab today, like we look at wolf ecology, we look at spider ecology, look at how wildfire is changing like soil dynamics. We looked at lake core sediments from June Lake. I mean, we've done like a lot of different things, and I think that's cool and fun to be able to think about all these different systems. But sharks came about my first year of grad school. I had friends that were going out and doing all this cool field work and doing stuff with isotopes and like horses and fur seals and dire wolves and all these cool things. And I was actually looking at groundwater, surface water of agricultural nitrates. So like the fertilizers that they put into agricultural fields in Corralitos, in the Central Valley or in the Central Coast. And I was like sitting there doing all this field work and I was like, this is terrible. Like, it's so smelly and gross here. Like, what am I doing? I should work on like some charismatic megafauna. And so then I was just like, sharks. There's a lot of shark teeth out in the world. Like, people know a lot about sharks. Like, sure, I'll do that. And so it was really just kind of this harebrained idea that I came up with. My advisor was very supportive and let me supported me in keeping sharks in captivity for my dissertation project. And then in Santa Cruz, there's like various organizations that work on white sharks and they do a lot of tagging of white sharks in the area. So, I mean, I just kind of like poked and prodded and started myself into a lot of spaces, but it was very much like, I wasn't part of a shark lab. I didn't know anything about sharks. I mean, the first time I watched Shark Week was actually like the episode that I was on. But like people love sharks. And so I sort of see it as like now a great conduit for getting people interested and excited in science. Or maybe even like, and to like slip in isotopes and be like, yeah, see, chemistry is cool. But it was like pretty random. It was not a clear path at all.
It sounds like the methodology kind of gave you that avenue into it.
Yeah, so I mean, grad students and even undergrads constantly are like, I love sharks. Like, I've loved sharks since I was 3 years old, and they like give me this whole like long story about how much they love sharks. And you're like, okay, like I might not be the person for you because I am basically a chemist. But some people sign on and then some people are like, no, I actually want to be like swimming with the sharks. I'm like, yeah, it turns out Merced, California, not so good for that. But it's really hard to get experience or get to work in a shark lab. Like it's very competitive. And very like white male dominated, and there's a lot of dynamics, you know, and power dynamics at play and all this stuff. And it's like, okay, if that's what you really want, like this is what it's gonna be for you. And that seems awful, but sure. And I was like, or you can do this backdoor way of chemistry, working in a lab a little bit. So yeah, it really was the method that drew me to sharks, but I also totally recognize that sharks are a great conduit for getting people interested in science now.
Did you have? Oh, no.
I have one more question. What do you think, the future of sharks is going to be with, like, I know there's a lot of like discussion about ocean temperatures rising, and do you think that there will be changes for them? I know they're also really resilient, but.
Yeah, that's a really great question. And honestly, for a lot of shark work, it's really behind on a lot of other organismal groups. Not really sure, I don't know why that is, but some of the other projections that are out there for like mammals or fish just doesn't exist for sharks. So that is an area of research that's pretty open for someone to pursue. I think we are seeing changes in shark distribution patterns already. There are reports of like in Santa Cruz, and then also the like Cape Cod, that there's a lot more juvenile sharks that are showing up than previously, and that there's these aggregations that are happening, which we never really saw before. And so there's definitely impacts that are happening. And I mean, some sharks might not do so great, especially given that their numbers are pretty low with various fishing pressures. I think they will survive as a group, maybe not all of them, but I think that they are very resilient. Like when I kept sharks in captivity, sometimes individuals would like go on hunger strike for like weeks or months at a time. And I was like, well, you cannot be part of my experiment anymore, little sharky. Like this is the whole point of the experiment, is for you to eat. But they cannot eat for quite some time and still, you know, survive. So yeah, so there might be some changes locally, but in terms of like a broader and larger context, I think that sharks will probably be fine. Thank you. Yeah. Is there any questions on Zoom, actually?
No, this is
Okay.
just for the recording.
Okay.
Yeah.
Okay. Just in
Thank
case.
you, absolutely.
Great. Thanks everyone for coming.
Thank
Thank
you.
you so much.